Today I will be talking about LTP, or long-term potentiation, which is the molecular biological mechanism behind memory. Okay, so first, what is LTP? LTP is a type of synaptic plasticity. Synaptic plasticity is the ability of individual synapses to strengthen or weaken depending on their use. There are two types of synaptic plasticity. One is LTP, or long-term potentiation. Another is LTD, or long-term depression. As the name indicates, LTP strengthens the synapse, and LTD weakens it. In a simplified sense, you can think of LTP as responsible for memorizing things, and LTD is about forgetting things. Before going through the details of LTP, let's first look at the key players in this process. First of all, let's look at the receptors that are involved. There are two main receptors responsible for this process, NMDA and AMPA receptors. Both are ionotropic glutamate receptors. Okay, let's break this down in two parts. Ionotropic means that the receptor is an ion channel, and glutamate means, of course, that the receptor responds to glutamate. Both AMPA and NMDA receptors get activated by glutamate. Glutamate is the major excitatory neurotransmitter in the brain, which means that it will depolarize the membrane and excite the neuron. When AMPA receptors are open, sodium will come in. When NMDA receptors are open, calcium will come in. Both sodium and calcium will depolarize the membrane. As I said, both AMPA and NMDA receptor open when glutamate is present, right? But NMDA receptors have more requirements. They open only when three conditions are met simultaneously. First, glutamate must be bound to the receptor. Second, membrane must be strongly depolarized. And third, glycine or D-serine must be bound. D-serine is a stereoisomer of L-serine. Only L enantiomers are used to make protein. And D-serine is made by a racemase. You might be confused at this point. Glycine is an inhibitory neurotransmitter, which means that it will inhibit depolarization. Okay, so here's a question for you. To open the NMDA receptors, glutamate and glycine is required, and the membrane should be depolarized. But glycine inhibits depolarization. How does NMDA receptor get activated at the first place? Think about it. Glutamate excites the neuron, while glycine inhibits it. The answer is, the rise in glutamate overrides the inhibitory effect of glycine. Glycine is present in the synaptic cleft at relatively low but constant levels. When a presynaptic neuron releases glutamate, its concentration rises sharply and locally. This spike in glutamate concentration, combined with the existing glycine pool, is sufficient to trigger NMDA receptor activation before the inhibitory effect of glycine takes full effect. Okay, there's one more thing to consider. In the inactive state, NMDA receptors are blocked by magnesium ion, so calciums cannot flow in. But when the membrane is depolarized, the magnesium block is removed, and now calcium ion can flow inside the cell. So here's another question. What if glutamate is present, membrane is depolarized, but glycine or D-serine is not present? What do you think? The answer is, calcium influx through NMDA receptors becomes less likely, but is not entirely impossible. Glycine or D-serine is a coactivator of the receptor. When glycine or D-serine binds to its own site on the NMDA receptor, it enhances the affinity and binding strength of glutamate. This increases the chances of glutamate binding and initiating the activation process. So, in conclusion, while glutamate and depolarization alone can theoretically open the NMDA receptor channel and allow some calcium entry, the lack of glycine or D-serine significantly dampens this effect. The resulting calcium influx is very small and insufficient for driving major downstream signaling cascades and LTP. So, now let's look at the very simplified process of LTP. First, glutamate released by activated presynaptic nerve terminal opens AMPA receptors, allowing sodium influx that begins to depolarize the postsynaptic membrane. Second, partial depolarization, and although not shown, glycine or D-serine is required. So when all conditions are met, the magnesium block is removed from NMDA receptors, which allows calcium to enter the postsynaptic cell. Third, increased calciums in the cytosol initiates various signaling cascades, and one result is that the postsynaptic cell will insert new AMPA receptors in the plasma membrane, increasing the cell's sensitivity to glutamate. We've been through the details of how NMDA and AMPA receptors work. Now, I'll provide a more general meaning behind this process. Neuronal signals are frequency modulated. This means that information is encoded not just in the presence or absence of a signal, but also in the rate at which it occurs. When the action potential reaches the end of the nerve terminal, the vesicles containing neurotransmitters that are docked in the presynaptic membrane will be released onto the synaptic cleft. In this case, the vesicles contain glutamate. The activation of AMPA receptor is very rapid, so when the firing frequency of the presynaptic neuron is high, the pulses of glutamate released is high, and the increased concentration of glutamate leads to more frequent binding to AMPA receptors on the postsynaptic neuron. High pulses of AMPA receptor activation lead to high-frequency voltage pulses in the postsynaptic neuron, and this, in turn, leads to the postsynaptic membrane potential to stay depolarized for a longer duration. This sustained depolarization, along with the bound glutamate and glycine or D-serine, creates the condition for NMDA receptor activation. The probability of the NMDA receptor channel opening and allowing calcium influx is not just a binary on or off state. It's graded and depends on the frequency of the voltage pulses. At low frequencies, even if the above conditions are met, the channel remains closed most of the time due to the magnesium block and relatively slow glutamate binding and unbinding dynamics. So, consequently, calcium influx is minimal. At high frequencies, the depolarization periods are long enough to allow more frequent glutamate binding and channel opening events, overcoming the magnesium block more effectively. This leads to a higher open probability and increased calcium influx. The more frequent the NMDA receptor is activated, the higher the frequency of the pulses of calcium influx, and this, in turn, is ultimately encoded as the pulses of downstream effector activity. So, in this manner, NMDA receptor activity critically depends on the frequency with which the synapse is used. So, you might wonder, how is this oscillations of calcium level ultimately interpreted by the cell? There are several calcium-sensitive proteins that translate the frequency of calcium influx into a wide range of biological activity. We'll discuss two major players in LTP and LTD, CAM kinase or calcium calmodulin-dependent kinase and calcineurin. As the name indicates, CAM kinases are calmodulin dependent. Calmodulin is a protein that gets activated when calcium is bound. Calmodulin itself has no enzymatic activity, but instead acts by binding to and activating other proteins. There are two phases of LTP. One is early LTP, and another is late LTP. In the beginning of LTP, that is, in the early phase of LTP, the effect is mainly exerted by the post-translational modifications, such as phosphorylation of pre-existing proteins. There is no de novo synthesis of proteins. You can think of early LTP as short-term memory. Late LTP is equivalent to long-term memory. In the late phase, new genes will be expressed. Aside from gene expression, there are changes in the morphology of neurons. That means remodeling of postsynaptic dendritic spines and formation of new synaptic connections will occur. Okay, now let's look at what would happen during the early phase of LTP. First of all, phosphorylation of AMPA receptors. CAM kinase directly phosphorylates AMPA receptors at various sites, increasing their surface expression and channel conductance. This translates to a stronger signal transmission at the synapse. Secondly, stargazin phosphorylation. CAM kinase phosphorylates stargazin. Stargazin is actually comes from a mutant mouse, named after the mouse, and a transmembrane protein that links AMPA receptors to postsynaptic density. So, this allows more AMPA receptors to be recruited to the synapse, further enhancing synaptic strength. And finally, NMDA receptor modulation. CAM kinase interacts with NMDA receptors, potentially influencing their channel properties and gating. This would contribute to sustained calcium influx needed for further LTP signaling. So, in late LTP, firstly, structural changes. CAM kinase would promote the growth and stabilization of synaptic spines, physical connections between neurons, and strengthening the synapse physically. Secondly, gene expression. When activated, CAM kinase will phosphorylate CRAB, and CRAB will bind to CAMP response element. A response element is a short sequence of DNA within a gene promoter or enhancer region that are able to bind specific transcription factors. CRAB regulates various genes, including ARC, CFOS, BDNF, tyrosine hydroxylase, numerous neuropeptides, and genes involved in the circadian clock. CRAB is often called as master of memory gene. And we have ARC, right? ARC is a very interesting protein. ARC mRNA is localized on the synaptic site and is translated there. So, during the initial stages of LTP induction, ARC can promote AMPA receptor endocytosis, leading to their removal from the postsynaptic membrane. However, this removal is thought to be a protective mechanism to prevent overexcitation and maintain synaptic homeostasis. In the later phase of LTP, ARC can also contribute to AMPA receptor stabilization and increase surface expression, enhancing synaptic strength. So, I'll tell you why I said that ARC is an interesting protein. It actually is thought to be originated from a retrotransposon, and really mind-blowing thing is that ARC can form a virus-like capsid and package mRNA and transports between the neurons. Isn't it amazing? Okay, this is the ARC capsid. And now, let's focus on calcineurin. Just like CAM kinase, calcineurin is activated by calmodulin. Calcineurin is a serine-threonine phosphatase. It opposes the action of CAM kinase. During early LTD, firstly, it promotes AMPA receptor internalization. Calcineurin dephosphorylates AMPA receptors, leading to their removal from the postsynaptic membrane and reduced synaptic strength. This weakens the signal transmission between neurons. Calcineurin can also dephosphorylate stargazin and thereby disrupting its role in AMPA receptor anchoring and promoting their internalization. Similar to its action on AMPA receptors, calcineurin can dephosphorylate specific sites on NMDA receptor subunits. This can alter the channel properties of NMDA receptors, affecting their open probability and surface expression. During late LTD, structural changes. Calcineurin is involved in modifying synaptic spine morphology, potentially contributing to physical weakening of the connection. And gene expression. These transcription factors are dephosphorylated by calcineurin. They play various roles such as regulating AMPA receptor trafficking, stabilization, endocytosis and degradation, and in many other aspects. CAM kinase and calcineurin is both activated by calmodulin, and calmodulin in turn is activated by calcium. At this point, you might wonder, how does cell differentiate between LTP and LTD? Although both process requires a rise in calcium level, there must be a difference, right? What could it be? What difference can be made? The answer is LTP and LTD depends on the level of calcium that is present. So it means when there is a repetitive, strong stimulus, then the NMDA receptor will be highly active and resulting calcium influx will be high. When the calcium level is high, CAM kinase gets activated and LTP occurs, and when the calcium level is weak, then calcineurin gets activated and LTD is induced. So, now let's look at how CAM kinase works. Each CAM kinase 2 protein has two major domains, an amino terminal kinase domain, the green one, and a carboxyl terminal hub domain, the blue one, which is linked by a regulatory segment. 6 CAM kinase 2 proteins are assembled into a giant ring in which the hub domains interact tightly to produce a central structure that is surrounded by kinase domains. The complete enzyme contains 2 stacked rings for a total of 12 kinase proteins, but only one ring is shown here. When the enzyme is inactive, the ring exists in a dynamic equilibrium between two states. The first, the upper left one, is a compact state, in which the kinase domains interact with the hub domains so that the regulatory segments are buried in the kinase active sites and thereby block catalytic activity. And in the second inactive state, the upper middle one, the kinase domain has popped out and it is linked to its hub domain by its regulatory segment, which continues to inhibit the kinase domain but is now accessible to calcium calmodulin. And if present, calcium calmodulin will bind the regulatory segment and prevent it from inhibiting kinase, thereby locking the kinase in an active state, the upper right one. And if the adjacent kinase domain also pops out from the hub, it will also be activated by calcium calmodulin, and the two kinase domains will then phosphorylate each other on their regulatory segments, the lower right one. This autophosphorylation further activates the enzyme. It also prolongs the activity of the enzyme in two ways. First, It traps the bound calcium colmodulin so that it does not dissociate from the enzyme until cytosolic calcium levels return to basal values for at least 10 seconds. Second, it converts the enzyme to a calcium-independent form so that the kinase remains active even after the calcium colmodulin dissociates from it. And this activity continues until the action of a protein phosphatase overrides the autophosphorylation activity of CAM kinase 2. At low frequencies of calcium spikes, the enzyme becomes inactive after each spike. As the autophosphorylation induced by calcium colmodulin binding does not maintain the enzyme's activity long enough for the enzyme to remain active until the next calcium spike arrives. And as you can see it in B, at higher spike frequencies, the enzyme fails to inactivate completely between calcium spikes, so its activity ratchets up with each spike. But if the spike frequency is high enough, this progressive increase in enzyme activity will continue until the enzyme is autophosphorylated on all subunits and is therefore maximally activated. Once enough of its subunits are autophosphorylated, the enzyme can be maintained in a highly active state even with a relatively low frequency of calcium spikes, which is a form of cell memory. The binding of calcium colmodulin to the enzyme is enhanced by calcium CAM kinase 2 autophosphorylation, so it is an additional form of positive feedback, which helps to generate a more switch-like response to repeated calcium spikes. So here are the references.